So in this presentation, we're going to go over DNA isolation. This is one of the important techniques you should know when you're working in the lab. And there are many different kinds of kits available from different companies, such as Kaizen, Invitrogen, and Promega by Basic. And these kits are the most popular one these days. And today I'm going to explain how to isolate DNA from animal tissue using Kaizen DNA kit, because I think this is what the most labs use these days. And even though you're using different kits, I'm sure the procedures are pretty much the same thing as this one. So even if you use other kits, you wouldn't have problem using it after this lecture. Now, just in case you don't know what the DNA extraction is about, I'm going to briefly explain about DNA. So we all have DNA in our bodies, but where can we find it? Basically everywhere, from your blood, skin, hair, just about everywhere. And we have about 100 trillions of cells in the body. So even from a small piece of skin tissue, we can find many cells. And that cell contains the DNA in their nucleus. So now that's what we're going for. We have to break it down and take the DNA out. So the first step of this procedure would be getting the animal tissue. For example, we can use the tip of the mouse tail. Many genetic labs use this for genotyping their rat. And they use about 0.5 centimeter, and they would get sufficient amount of DNA out of it. So you place this tail into 1.5 milliliter microcentrifuge tube and add 180 microliter of buffer ATL and 20 microliter of proteinase K. ATL is tissue lysis buffer, and it is used to induce lysis of cell tissue. And proteinase K digests the proteins, and it will rapidly inactivate the nucleus that might degrade our DNA during purification. So after vortexing this tube, it has to be incubated at 56 degrees Celsius for about 6 to 8 hours when you're digesting mouse tails. So this lysis time could vary, depends on the type of tissue you're using. After lysis step, vortex the tube about 15 seconds and add 200 microliter of buffer AL and 200 microliter of 100% ethanol. And then you will have to vortex it again. Buffer AL is cell lysis buffer, so this step will break down the cell membrane and all the DNA will come out from the nucleus. The ethanol will help separate the DNA from the other components such as protein and RNA. And then we need to pipette the mixture into DNA's mini spin column placed in 2 ml collection tube and centrifuge it at 8,000 RPM for 1 minute. The spin column has a unique silica gel membrane at the bottom that binds up to 20 microgram of DNA in the presence of high concentration of chaotropic salt. And rest of the junk will pass through the filter, so we can just discard the flow through. Then we need to perform 2 washing steps to get rid of the other cellular compartments that might have attached to the membrane. First, we will add 500 microliter of AW1 buffer and spin it for 1 minute at 8,000 RPM. And then do it one more time with 500 microliter of AW2 buffer and spin it for 3 minutes at this time and at 14,000 RPM. At both steps, the flow through needs to be discarded. Now we can elute the DNA that has been bound to the membrane using elution buffer, which is labeled as AE on the bottle, and add 200 microliter on the spin column. On this step, we need to place the spin column into a clean 1.5 milliliter microcentrifuge tube. As we did before, this will be centrifuged for 1 minute at speed 8,000 RPM. To increase the DNA yield, we can separate the step into 2 by using 100 microliter of elution buffer each time. So if you did everything correct, you should have around 20 microgram of DNA in the tube, and you can proceed with genotyping. So this is the whole step of purifying the total DNA from animal tissue.